Well, I graduated with a degree in physics and engineering, and while I was really impressed with the elegance of the physics that I was studying, I was more impressed with the diversity of biological questions that were available to be answered. Just a completely new field was emerging, and it looked to be a really exciting thing to be part of that. At the same time, I learned that my father had been diagnosed with terminal lung cancer, and I was really depressed to learn what few treatment options we had for terrible diseases like that. And so I really became interested in trying to understand how one might take some of the expertise that I'd learned in physics and bring that to bear in the biological sciences to both participate in some of these new directions that were emerging biologically, and also hopefully in the long run to be able to move in the direction of doing something that would benefit mankind, and in particular to move us in the direction of thinking about how to do cancer treatment better. One of my research interests is in trying to improve our ability to detect cancers that are prone to metastasize early enough that we can detect them before they have gone through the metastatic process. I've been involved in a broad range of scientific research over the years, and actually this has resulted in the issuance of over 60 patents at this point. But you've asked me which ones are the most important, and I think that there are two patents that sort of crystallize research that has had a major impact on breast cancer already. One of these had to do with the development of the ability to visualize individual DNA segments within cancerous cells. And what this allows us to do is to look for things like gene amplification. You literally look in the microscope and see these amplified or sequences that are present in too many copies. Now, one class of such abnormalities is HER2 amplification. And so what we've done is to develop a diagnostic test that is now used routinely throughout the world to help classify or understand which are the HER2 positive tumors that are going to be treated with drugs like Herceptin or lapatinib and so on. So participating in that development I thought was really quite exciting. So the other technology that we've developed, called comparative genomic hybridization, allows us to look not just at one part of the genome, but it allows us to look at the entire genome and to discover which are the regions of the genome that are recurrently gained, those that are recurrently lost, and what that has allowed us to do is to identify new therapeutic targets and given us new insights into how these abnormalities contribute to the pathophysiology of cancer. And so I think it's taken us a major step forward in understanding what actually goes wrong in these cancers. And one of the really remarkable things that's come out of that is how much difference there is from cancer to cancer. In fact, I would say that having looked at thousands of cancers at this point, it's probably true that we've never seen two that were exactly the same. So this has given us a very important insight into why it is that some of the nominally similar clinical cancers actually respond quite differently to the therapeutic approaches that we bring to them. They are just genomically different. I think that there are a number of exciting developments in the field of breast cancer research today that are going to really move us along rapidly in the development of personalized cancer treatment. One of the things is the technologies that allow us to analyze what's gone wrong in cancers at the DNA level, at the RNA level, at the protein level, and so on are evolving at light speed. 10 years ago, the cost of sequencing a cancer genome would have been in the tens of millions of dollars. Today, it's more like the thousands of dollars. And so what this gives us is a tool to give an almost perfect fingerprint of every cancer. And so this is going to allow us to know in exquisite detail what's gone wrong with each cancer. So I'm really honored to be receiving this recognition from Zero Breast Cancer. What that means to me is that we have been successful in engaging the advocate community in our research program. I think that's critically important. I believe that the advocates have played roles in a variety of ways. in helping us move our research programs forward. The advocates keep pushing us in the direction of making sure that we are focused on real problems that are important to the patients, and perhaps have gotten us away a little bit from having to do the next most elegant paper that would perhaps show up in Science. So, it's given us that motivation to go forward on the hard problems.